Welcome! Today we are diving headfirst into the next generation of gene editing. We're talking about a world where we're not just fixing tiny genetic typos, but potentially rewriting the entire rulebook for how we fight disease. It's a huge leap forward. But first, let's start with a number to really set the stakes. 10 million. That's how many people are projected to die every single year from antibiotic-resistant superbugs by 2050. It is a massive global crisis that's literally racing towards us. And right here, this is the bottom line, the big takeaway right up front. What if we could not just fight back, but actually reverse that resistance? And at the same time, what if we could dramatically boost the power of our best gene editing tools? That's the core promise we're going to unpack today. Okay, so to really get a grip on these incredible upgrades, we first need to understand the original tool that started it all. So let's do a quick refresher on what CRISPR is and how it works. The easiest way to think about CRISPR-Cas9 is like a find and replace function, but for the code of life itself. It's this incredible molecular machine that scientists can program to go into a cell's DNA, find a very specific sequence, and make a precise change. It's unbelievably powerful. So how does it actually do it? Well, it's a pretty straightforward three-step process. First, you've got the find stage. A little molecule called a guide RNA acts like a GPS, leading the whole system to the exact spot on the DNA strand. Then comes the cut. An enzyme called Cas9 acts like a pair of molecular scissors and makes a clean snip. And finally, replace. The cell's own natural repair crew comes in, stitches the DNA back together, and, this is the clever part, it can use a new template provided by scientists to change the code. And I want to be super clear here. This is not science fiction anymore. Late last year, regulators in both the UK and the US gave the green light to the very first therapies based on CRISPR. They're being used to treat devastating inherited blood disorders like sickle cell anemia. It's real, and it's already changing lives. But, science never sleeps. So now we get to the really exciting part, the reason we're all here, the next-gen upgrades that are taking CRISPR's potential into a whole new stratosphere. We're going to look at two powerful new tools. All right, upgrade number one is a total game changer for that superbug crisis we talked about. This isn't about making a bigger, badder antibiotic to kill bacteria. No, this is much smarter. It's a system designed to sneak inside the bacteria and basically strip away their defenses, leaving them totally vulnerable to the antibiotics we already have. And the efficiency of this thing is it's mind-blowing. Listen to what Professor Ethan Beer from UC San Diego says. He explains that with this method, you can take just a few cells, release them, and they will go on to neutralize antibiotic resistance in a huge population of bacteria. It's basically a self-spreading solution. This chart really lays out the difference. Think about it. Standard antibiotics are like a sledgehammer. They just try to kill everything, which actually helps resistance grow. And they can't get through these protective shields bacteria build called biofilms. This new CRISPR system, though, it's like a surgical strike. It spreads like a software patch, disabling these specific genes for resistance from the inside. It can even get through those biofilms and reverse resistance in the whole colony. Okay, let's move on to the second major upgrade. This one solves a totally different but equally huge problem: efficiency. How do you get the gene editor to enough cells in the human body? Well, the concept here is just brilliant. What if you could deliver a gene editor that makes copies of itself and spreads to neighboring cells? Kind of like a good virus. It's called the NANITE System, and here's the big idea. You treat just an initial group of cells. The NANITE system then hijacks that cell's own machinery to build more copies of the entire CRISPR tool, packages them up, and then sends them out to treat all the surrounding cells. It's an amazing way to solve the numbers game because it amplifies itself.
And the results really do speak for themselves. Check out this chart from a mouse study. They were targeting a harmful protein in the liver. With the same dose, you can see standard CRISPR only managed to edit about 4% of the target cells. But the NANITE system, because it can spread, hit 11%. That's nearly a 3 times bigger impact.
Now, as exciting as all this is, we've got to bring ourselves back down to earth for a reality check. Because moving these incredible tools from a lab into a treatment you can get at the doctor's office, well, that means overcoming some really significant hurdles. There are really 3 main challenges on the road to the clinic. First, you have off-target effects. That's the risk that your molecular scissors make a cut in the wrong place. Second is the delivery problem. This is the massive logistical challenge of just getting the tool to the right cells in the body. And third, immunogenicity, which is just a fancy way of saying there's a risk your own immune system will see the CRISPR tool as an enemy and attack it. Let's just focus on that delivery problem for a second, because it really highlights the complexity here. Think of it like a super specialized shipping challenge. You have this very fragile package, the CRISPR machinery, and you need to get it to a very specific address, like only your liver cells, without it getting lost or destroyed along the way. Right now, scientists often use deactivated viruses as little biological delivery drones to carry that package, but it's a huge area of ongoing research. So when you put it all together, you've got these unbelievable breakthroughs on one hand and these very real-world challenges on the other. What does the road ahead actually look like for our medical future? This table kind of puts it all in perspective. For the superbug crisis, we now have this potential solution in the CRISPR Gene Drive. For the problem of low editing efficiency inside the body, we've got the self-spreading NANITE System. And ultimately, both of these breakthroughs push the entire field of genetic medicine forward by making it safer and giving it a much wider reach. So what's the big takeaway here? Yes, the hurdles are absolutely real. We can't downplay them, but the pace of innovation in this field is just extraordinary. These aren't just ideas on a whiteboard anymore. We're seeing tools move from a cool theory to proven results at a truly breakneck speed. And that leaves us with one last big thought. As we gain this unbelievable power to literally rewrite the code of life, the question really isn't, can we do it anymore? The question is becoming, what should we do first? With tools this powerful, the future is going to be fascinating to watch.